Now we talk about gene prediction. So gene prediction involves the prediction of coding sequences and predictions of where the translation starts. Since we are actually corresponding the genes with the proteins, so we are interested in finding the translation starts, which is actually the it starts with ATG. Then we also predict the prediction of splice junctions. In case of eukaryotes, we know that exons and introns, their boundaries are marked by these splice sites or splice junctions. What is the coding sequence? The portion of genes DNA or RNA that is composed of exons and that codes for protein. So exons put together, they make up the coding sequence. So normally the coding sequence starts with start codon ATG and it ends with one of the three codons, which are the stop codons. This region is bounded nearer 5' end by start codon and 3' end by stop codon. Any full messenger RNA that is obtained from a gene that can have a CDS. So normally, in order to get the coding sequence or CDS, sometimes we get the RNA, messenger RNA, and we kind of make it its DNA copy, cDNA. That process is called as reverse transcription. What is an ORF then? So open reading frame or ORF is the sequence. We can get any DNA sequence and we start looking for ORF, where ORF is defined as anything that is bound between a start codon and one of the stop codons. So that might contain regions other than exons. So these are actually the predicted sequences. Since we are talking about those codons, the triplet codons, here is how it looks. This is the codon table. So our first letter is U. Sometimes it is written with U's and some literature you would find with T, but never mind. U is T and it's T is U in messenger RNA. So we can have U in one side and we can have U here and we can have U here. So in this way U, and it encodes into phenylalanine. How do we get into it? We try different combinations. So since there are 20 amino acids and there are 4 nucleotides, so if we get a sequence of one nucleotide like A, T, G and C, we might end up representing only 4 amino acids. So what if that codon, like the sequence of amino acids is determined by these codons? So what if we assume that codon is made up of 2 nucleotides, like for example A with rest of them, A with A, and we can have A with C and so on. So in this way we can have 16 possible combinations. But here our amino acids are still more than that, so they are 20. So they thought the molecular biologists, they came with an explanation that this actual codon is actually made up of 3 nucleotides. So in this way we will have 64 codons. Obviously all of them are not coding. There are some stop codons, and there is obviously this start codon here, AUG, ATG in DNA, and so on. We have those stop codons here, 2 of them here in this window and 1 of them is here. So 3 stop codons, and there may be more than 1 codons for some amino acids. So there comes the concept of degeneracy of this codon. So it's degenerate, so there can be more than 1 codons. So how CDS and ORF, they differ from one another? CDS is transcribed and is coding for something, whereas ORF, you can get it through predictions. So all ORF may not be coding. So you get those ORF open reading frame by simply defining or simply designing an algorithm where you can ask your program to look into some ATG and some in-between sequence and stop codons. What are splice junctions? So we have introns in eukaryotes. So introns and exons, they are separated from one another, but at the junction of an exon and an intron, we start with GT. So actually this is the segment that is part of intron. So introns, they start with GT and they end up with AG. So if we have GTs and AGs and they are spaced by certain distance, we suspect that this region is actually an intron. So the presence of these splice junctions, we call donor and acceptor because of their behavior in splicing. I'm not going to cover that. but they are called as donor site and acceptor site. So these donor site and acceptor site, their prediction is also important in gene prediction. Here is how it looks like in the actual sequence. So you see those splice sites are shown in red here in this diagram, introns are black and exons are blue. So we have GT and then we have a big intron and that ends up with AG, and then exon starts, and then with GT another intron starts and then ends up with AG. So in this way these introns and exons are separated from one another, and we can get their boundaries by looking into these splice junctions. This is how it looks like in prokaryotes. We have genes which are actually transcribed from same promoter. So, and every gene within that we can get a start and a stop codon. But these genes, they are transcribed from, as I said, from the same promoter. Whereas in case of the eukaryotes, here we are focusing on only one gene. So there is a promoter, and then obviously there is some intron region here, and we have exons. So exons are separated from one another through introns, and we can have a start site and for that exon. So actually the start for getting that protein, this translation start site and stop site in the last exon. So first exon has start and the last exon has stop, and introns are, they are marked by these donor and acceptor sites. So pretty complicated situation as far as the eukaryotic organisms are concerned. We combine different pieces of information when we get the idea about these start sites, and we get the idea about UTRs and different splicing variants and these donor and acceptor sites. We put them together. So this thing is called as information fusion. So we get different pieces of evidences while looking into these different patterns, or we can call them as signals. And these signals, they help us identify the genes. What is the signal and what is the content? So we use these terms most often when we are doing gene prediction. So signal is a small pattern within the DNA, whereas the whole region of the genomic DNA can be called as a content. So for example, splice sites, starts and ends of transcription or translation, branch points, these all things, they are called as signals, whereas contents may be the exons, introns, UTRs and the promoter regions. So promoter regions, they have some specific patterns in them. So that pattern is called as a signal, whereas the whole promoter can be called as a content. So gene prediction involves prediction of coding regions, start sites, as well as splice junctions.